Thank you, Peg, and all the panelists this morning. So for the next 15 or 20 minutes, we're going to moderate a discussion among our three panelists, and then we'll open up to general questions from the floor for the following 20 minutes or so. So just to get us started, we're lucky today to have a number of clinicians in our audience, and I'd love to hear from each of you what you'd like clinicians to take home from each of your talks, what your take-home messages are for them. Marty, go ahead.
I'll start because I'm a clinician as well.
Exactly.
I say my take-home message is that for 50 or 60 years we've been giving antibiotics a blind eye. We haven't assessed that there is cost to giving antibiotics, cost beyond resistance. We've known about the resistance cost for some time. But we have to start discovering exactly what those costs are in terms of our ecosystem and the clinical manifestations, and start including that into the decision whether to treat or not. And countries are different. In Sweden, they're using much less antibiotics than we are. They're using about 40% of the antibiotics we do in clinical medicine at every age, in childhood and adulthood. So they're recalculating differently. In the United States, there's tremendous variability in antibiotic usage. So it's achievable once we start paying attention.
Excellent. Thank you. Gautam?
So I'll perhaps just repeat something I said earlier during my remarks, and that is, as a physician, to consider that the future of clinical studies take into account this idea that we have these unique personalized ecosystems which are hit every time we give any kind of drug. And antibiotics are what we speak about now, but essentially any chemotherapeutic that you throw towards the human body has this option and high likelihood of perturbing the microbial ecosystem. And so those of you who sit on IRBs and who think about the design of clinical studies, we've been hearing about personalized medicine from the fact that we have unique individual genomes, but now to consider that we actually have unique individualized pangenomes that take the microbes into account.
Right.
I guess I would urge you to advocate for better diagnostics. I think the reason we have to rely on antibiotics so often is because we know so little about what the infections are that we're dealing with. I can tell you from my own personal experience that if I have a urinary tract infection, nobody bothers to culture it. They just give me the latest standard antibiotic, or they did until 10 years ago when I stopped taking them. We've got to find a new approach to figuring out who's involved in these multi-species infections. There is no magic bullet on the horizon, no gorillicin sitting out there waiting for us. We have to have a new paradigm, and the only way we're going to have a new paradigm is by knowing who the players are at the species level.
So what's in the way, you think, of actually developing that sort of approach to diagnosis? So in the President's executive order, on drug resistance, he had made a directive to set up a $20 million prize for molecular diagnostics. Is that sufficient? What else has to happen to really push this forward?
Well,
an asset of all the panel, but Peg, go ahead.
Well, one of the outcome of the 9/11 devastation and the subsequent anthrax scare was that scared us into diagnostics, and we spent billions of dollars beginning that process of developing diagnostics. Now, that's not a field I'm familiar with, but I can tell you right now, we still don't have the bedside diagnostics that we all thought we would have by now. So maybe we need to add a few zeros.
I think like many aspects of American medicine, the cost model is all wrong. We think that giving an inexpensive antibiotics is a low-cost solution, but in fact it may be a very high-cost solution. And once we calculate the cost, then it will become apparent that having good diagnostics will be cost-effective.
Absolutely.
Right now it's not, because we have this umbrella of inexpensive antibiotics that are given out like water to 70% of the children who come in with respiratory infections, even though we know that most of them are viral. But no one is quite ready to bite the bullet on their child or their patient. Or again, there's a lot of variability. So we have to cost it out. Once we do the cost, improved diagnostics will be part of it, so that we know who's got a viral infection, who's got a bacterial infection, and if it's a bacterium, which one, so that we can take out our narrow spectrum agent and treat it, if needed. Not every infection needs to be treated.
right.
That's yet another point of our under-costing what antibiotics do.
I'd say one of the barriers that, as a young faculty member, that I perceive is much like this rhetoric of warfare. We're stuck, I think, in our funding system for research in the rhetoric of translation. And I think that's wonderful. We critically need translation research, this idea from bench to bedside. But I think what we sometimes forget is the only reason we can do anything at the bedside is because of the toil at the bench. And so with needing new diagnostics, I firmly believe that we need a huge focus on the basic science aspects, these questions of microbial ecology and which bacteria are out there and what they're doing. And if we lose our focus and jump immediately to try to only have translational goals, we won't have anything to translate in 10 or 15 years.
So can I add on to
Please.
that? So I stopped submitting NIH grant proposals probably maybe 10 years ago because the review panels would reply, why would we want a narrow spectrum drug? Nice proposal, pal, well written, good experiments, but why would we make that important? And so we have to open our minds to the fact that antibiotics are not the deal that they used to be, and we've got to look for alternatives, whether you like bacteriocins or phage or whatever. We've got to have NIH panels recognize that radical thinking is a good thing right now.
You know, as Peg mentioned, broad spectrum antibiotics will always be necessary for some terribly ill patients. But because the whole emphasis in antibiotic discovery has been on broad spectrum, one antibiotic that will affect all cell walls or all ribosomes, that's been the whole push of the pharmaceutical industry. Obviously, if you target single agents, the market is that much smaller. But it's a public good. We have to develop it as a public good, just as we build highways together as a public good. No one can afford the highway. But we need to use tax dollars in one form or another to create this public good.
We also need to impact the FDA because the hurdles that are placed for developing an antibiotic are just outrageous. They're unreachable in terms of producing clinical studies with meaningful numbers. It's very hard to do, and you can't blame the pharma industry for that. They didn't create that. Those are the hurdles they have to jump over. They're expensive, and they're almost impossible to reach.
One thing I'll add, and this is almost just an example of another strategy, at least what some in the audience will maybe have heard of, is in terms of developing narrow spectrum agents, again thinking of this from this microbial ecology perspective, is to consider going after the virulence factors in the bugs that cause disease. And so we usually think about this destruction model, where we go after key processes that keep the cell together, and there's always going to be a selection pressure there for the few resistant mutants to evolve and sweep the population. And so there is now preliminary work and some small biotechs looking into this, but maybe we go after the very mechanisms that allow these otherwise good, maybe dormant bugs to cause our diseases. And so that's just another parallel narrow spectrum therapy which may not have the same types of collateral damage.
And the genomic information available fuels this, makes this possible. So for every pathogen, we can find genes that are unique for that pathogen that no one else shares or nobody closely shares.
Excellent. Let's change gears for just a moment. But all of you have mentioned that the risks associated with antibiotic use and their solutions are bigger than a hospital or clinic. They involve more than just human-associated microbes. So I'd love to hear your perspectives on how to broaden this conversation about antibiotic use and risks and mitigation beyond hospitals, clinics, and the microbiome. How's that for?
I'll take a stab at that.
Thank you.
And I agree, we need to engage with the FDA and the USDA. I mean, you've heard this stat, a couple of us said it already, but you should just keep telling yourself that and tell your friends that 80% of these life-saving chemotherapeutics are being dumped to give us slightly less expensive meat. And I just don't think there's any reason for us to tolerate that. We should not just ban antibiotics, we should promote research to think of alternatives to allow animals to get bigger and more productive. Absolutely, we depend on that industry, but I think in terms of extending it beyond the clinic, where there are problems, there's the inappropriate use of drugs. But the fact that you've got the lion's share, the numbers speak for themselves. We need to do something about this wanton use and abuse, I would say, of antibiotics for what I think is a sort of ridiculous purpose.
And we have to also recognize that we're well past the point where there's a question about whether our antibiotic use outside of the clinic has an impact on humans. Duh. I mean, right? As Gautam would, if we gave him another half hour, he'd tell us that most of these resistance genes are 2.5 billion years old. It wasn't from us dumping antibiotics out there. They're out there doing things that have nothing to do with antibiotic resistance. But the microbes have evolved an enormous diversity, whether you're in a cattle field or the outback of Australia or a deep drill into the Antarctic, you're going to find antibiotic resistance genes. We don't know what they do. How can we not know with NIH? How can we not have funded those studies? I don't know about you, but it gets depressing trying to find the money for those studies. But regardless, they're there, and they are available to pathogens that make it into humans. So let's stop arguing about where did they come from. Doesn't matter, they're there. So.
And we've selected them.
Yes.
And the key word here is selection. And for my part, that's why I wrote Missing Microbes, is to bring this discussion to the general community, to the patients, to the parents, because we have all believed that antibiotics are miraculous drugs, and they are, that have no cost. And once parents begin to think maybe there's a cost, and if the evidence continues that maybe there is some incremental cost that their child, for every antibiotic course they take, will be more likely to become obese, as a study in JAMA Pediatrics suggested 2 weeks ago, and a study from Manitoba, and our own studies, cohort studies, suggest as well. If there's incremental cost, then parents are not going to push the doctors to give antibiotics. They will be relieved if the doctor says, I've examined your child carefully, and your child isn't that sick. They don't need an antibiotic. That parent will be relieved rather than feel deprived.
We're here in Washington, D.C., so it's hard for me not to think about all the action at this level last month, at the President's executive order, the PCAST report, et cetera, et cetera. So given all of that attention and perhaps momentum, the task force and directives, et cetera, what else has to happen, in your opinion, at the federal level? I mean, do we have a good first step now with all of this attention last month? Or are there other things that could happen at the federal level?
Are we all too jaded to reply?
Lots of things are going on now.
Yes,
And the question is that, Ebola is a terrible worldwide disaster. It's gotten enormous attention, appropriately so. This is a slower epidemic, but it's an important one. And in fact, work going on from the 1950s shows that antibiotic treatment increases susceptibility to pathogens. Mm-hmm. What's called colonization resistant, as we're all aware of. And so that's yet another thing. The overuse of antibiotics makes our population more susceptible to epidemic agents of any kind.
I guess I want to be an optimist, right? So I guess the idea that there are companies in Beijing or in Portland, Maine, or in New Zealand that are interested in these alternative approaches now, that's a good thing. It needs to happen at a much higher rate for us to succeed at this, because we know how many drugs fail before they make it to market. But getting an awareness that antibiotics are not the only solution. We need them desperately, but we don't need them for everything. And just reminding ourselves of that and using your purchasing dollars in that direction, antibiotic-free hamburger meat, for example. I mean, you guys may not know this, but bacteriocins, my protein, are used by McDonald's on salads. So you're all using my drug without even knowing it. So we got to get used to the idea that there are other ways.
And I echo this idea of the other way. One of us, or more of us may have said this already, but what do we do going beyond these federal reports that come out? And I think, using this language of communities across regulatory agencies as well, learning from our microbes again, that, as I said, we probably will never be successful, at least here with the current lobby structure, to immediately ban all antibiotics in food animals. But one engagement, a bone that we can throw out is research funded by the government on alternatives to antibiotics for growth promotion. Yeah. And if we can get enough people energized about that, enough researchers funded to do that, I think then we have an opportunity to tell the animal industry that we actually care about that problem and then incentivize them away from using antibiotics.